In this video, we're going to survey the classes of biomolecules that we're going to encounter across our studies of biochemistry. So let's dig into that. I think for a lot of students, when they first encounter biochemistry, they're overwhelmed by the details, right? So this is a poster showing a bunch of metabolic pathways. And I think a lot of students say, how in the world am I ever going to come to grips with all of this complexity? How am I going to pass a class where I have to deal with all of this complexity? But fortunately for us, actually biochemistry has some underlying modularity and simplicity that makes it more straightforward than you might think to try to understand these networks. And so this starts at the level of the number of atoms. So if you've taken a general chemistry class, you probably learned about all kinds of strange inorganic atoms and all kinds of complexity there. But it turns out in biochemistry, we can almost exclusively focus on only 6 atoms, which I'm showing here ranked by their commonality. So oxygen, carbon, hydrogen, nitrogen, phosphorus, and sulfur. That's in rank order. Those form 97.4% of the mass of a human being. So that's great because we have to spend much less time worrying about like rubidium or something than we would maybe if we're doing other sorts of chemistry. Further, if you want to remember what these are, I like the acronym CHNOPS, carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur, because it makes more English sense maybe than whatever the top one is. So we've got the small number of atoms, but further, this small number of atoms is actually arranged into a small number of core molecule types. And there's really only 4 that we have to worry about. Those 4 are lipids, which basically consist almost exclusively of carbons and hydrogens, with some oxygens decorating, but almost exclusively carbons and hydrogens. If we add a bunch of oxygens, we get to carbohydrates, which have carbons, hydrogens, and oxygens in them. If we add some nitrogens, we get to amino acids, which now have carbons, hydrogens, oxygens, and nitrogens. And if we add a bunch of phosphorus, we get to nucleic acids, which have carbon, hydrogen, oxygen, nitrogen, and phosphorus. And so what we're seeing here is that, yeah, there's a lot of complexity in biochemistry, but it's really built out of a small number of parts that are assembled together. Now you might be saying, well, wait a second, where's the sulfur? And you're right, sulfur appears in a few different amino acids. So that's the other place that we get the S in the CHNOPS. Okay, so small number of parts put together into a small number of molecules. Where does the complexity come from? And it turns out the complexity comes because these small number of parts are assembled combinatorially. So what do I mean by that? Well, imagine a DNA chain. So hopefully you've come across in the past that DNA is made out of bases, and those bases are A, T, G, and C. And if we imagine a chain of DNA, right, we have at the first position, we have a possibility of any of those 4 bases. At the next position, we have the possibility of any of those 4 bases. At the next position, we have the possibility of any of those 4 bases. So if we think about what are the possible nucleic acid chains or the possible DNA chains, it's 4 times 4 times 4 times 4. And it actually turns into 4 to the n, where n is the length of our chain. So if we have a nucleic acid sequence with 100 bases in it, there are 10 to the 60th possible DNA chains. So there's just an astronomically large number of chains. That's more than the number of atoms in the visible universe, right? So we can build enormous complexity from a very simple set of building blocks once we start doing things combinatorially. And we see that same basic phenomena happening when we're talking about amino acids, when we're talking about carbohydrates, and when we're talking about lipids. So a lot of the complexity comes from just combining a bunch of simple parts. So this puts us into what you might think of as a glass half full, glass half empty scenario. If you're a glass half empty kind of person, you're saying, I have to keep track of a ton of stuff to understand biochemistry. And that's true. There is just a ton of stuff going on that if you want to be successful in either a biochemistry class or as a practicing biochemist, you just have to keep track of a lot of details. But the glass half full kind of person is that if you understand a few core things well, then you're well positioned to understand the complexity and where that complexity comes from. And the idea of this video and sort of my approach to teaching biochemistry is to try to focus on that half full stuff. What are the core patterns, the core molecules that we have to understand if we're going to make sense of the amazing complexity of biochemistry? So our goals for today would that at the end of this video, you would be able to recognize the different molecular classes. So if you look at a big metabolic network or you look at a textbook, you'd be able to say, oh, look, that's a lipid, or oh, look, that's a carbohydrate. Second would be that you'd be able to reason about the sorts of molecular interactions that each of these molecules can form. And that actually pairs with the next video. So the next video, I'm going to talk about specific sorts of things like the hydrophobic effect and hydrogen bonds. So sort of with these two videos paired together, hopefully you'll be able to reason about, oh, like lipids like hydrophobic interactions and amino acids really like hydrogen bonds, for example. And the final goal is a negative goal. It's to not get lost in the details, right? There's a lot of stuff that we're going to cover even in this one short video. And the idea I'd continually be thinking, how do I sort of get my head up and just think about the patterns I'm seeing rather than worrying about a bunch of specific details, at least for now. Because once we get into future videos or you start going deeper into biochemistry, you're going to come across very specific detailed things that matter. From here, think of this as the broad brush overview that sets you up to be successful once you dig into a bunch of specific details. So, all right, with that in mind, let's start tackling this. What we're going to do is just work through each of the 4 molecular classes, talking about the features of these molecules and what they do. So we're going to go through lipids, carbohydrates, amino acids, and then nucleic acids. So lipids first. Okay, lipids are actually interesting to define because there's not a simple formula. I can't say, oh, a lipid will always have this ratio of carbons and hydrogens or something, or this stoichiometric formula. Rather, I think the simplest way to define lipids is actually metabolically. So a lipid is a hydrophobic molecule. Clearly did not leave myself enough space there. But it's a hydrophobic molecule where most of the carbons come from acetyl CoA. So this is a metabolic definition. We're saying it's hydrophobic and its carbons come from acetyl-CoA. What in the world does that mean? Well, let me draw an acetyl-CoA just to make your brain explode. So this is an acetyl-CoA. It's this big, complicated looking molecule, but it turns out all of this stuff is basically a handle for moving around these 2 carbon units, carbon 1, carbon 2. And so acetyl-CoA is a critical molecule that's used across metabolism to deliver carbons to other places. And so what we can see if we map out the sort of synthesis of lipids is that we start with an acetyl-CoA and it donates 2 carbon units, or it actually takes some other numbers of carbons, but 2 and a little bit more carbon units to go and build out a branch tree of possible outcomes. So it goes to this intermediate, which branches out to this intermediate, which branches out to this intermediate. If we go up to the top, we've got all these complicated places it can go. So a lipid, again, is a hydrophobic molecule that's built out of this acetyl-CoA network. Okay, great. There's a lot I could talk about here, but I'm actually going to only focus on 2 lipids for this video because this actually forms the bulk of the stuff you have to worry about in most of biochemistry. And those are fatty acids and cholesterol. So we're going to go through each of those classes of molecules here in this survey. All right. What is a fatty acid? A fatty acid is something that has some fatty parts and some acid parts. And so it's a carbon chain. In this case, I'm drawing a C16. So this is a 16 carbon acyl chain that is attached to a carboxylate or carboxylic acid if it's protonated. So this is the carboxylate. What I'm showing here is the fatty acid known as palmitate, which is, I guess, the 16 carbon version. This is the most common fatty acid in humans. Now, you might be looking at this and saying, well, wait a second, you listed it as palmitate and as hexadecanoic acid and as the C16.0. I'm putting those names on there just so that you'll know that with fatty acids, there's actually multiple levels of nomenclature. So palmitate is what this is normally called because it's such a common fatty acid. But if you look across papers or Wikipedia or other places, it might be listed as different names. So I'm putting those out there for completeness, not to get worried about the details. Okay, so let's talk about the molecular structure a little bit. So it's got this negatively charged thing that can interact with water, and then this really long unsaturated acyl chain. That unsaturated acyl chain has lots of degrees of freedom, right? Each of those bonds can rotate. So this is a pretty flexible, floppy molecule. But it really, really does not like water, right? Because it's got this long, greasy hydrophobic chain. Fatty acids are used for a variety of different functions. Maybe primarily we'll think about them in this set of videos, at least, as a fuel source. There are many cell types that actually really prefer fatty acids to other fuels. They're also used for energy storage. You can take 3 fatty acids and glue them together to make triglycerides, which maybe you've heard of before. That's how we store fat in, say, fat cells. And then finally, they have signaling roles where different fatty acid versions can go around and can have different signaling outcomes. Now you might be saying, wait, different fatty acid versions? What do you mean by that? Well, if you think about this chain, there's actually quite a bit of things that you could vary The two major things that get varied biologically are the length and the saturation of this chain. So I'm showing here, this is like I said before, palmitate is the most common fatty acid in humans. It's got 16 carbons, but there's other ones. So this is a carbon that only has a fatty acid that only has 14 carbons, otherwise identical to palmitate. There are other fatty acids that are longer than palmitate, right? So you can vary the number of carbons in the chain. You can also vary the number of double bonds. And so this fatty acid here is a C16 fatty acid, but it has a single double bond in it. So when you start looking across biochemistry, what you'll start encountering is, well, there are fatty acids, and then there are fatty acids that have different chain lengths and different double bond character. And once we get into fatty acid metabolism, we'll start taking that apart in more detail. Okay, so that's fatty acids. Let's talk about cholesterol. So remember, these are both related to each other in that they both come from acetyl-CoA, so they're both built out. They're hydrophobic molecules. What you might notice when you look at cholesterol is that it's chunkier. It's got more interesting structure than a fatty acid. So unlike a fatty acid, which is just a long acyl chain, this actually has assembled rings. The rings are always there, and that it's got 4 of them that are so common that they have names, A, B, C, and D. And hopefully what you appreciate is that there's a whole bunch of carbon on here and that there's just one oxygen, one hydroxyl that hangs off of the A ring. Cholesterol, because it's big, bulky, and hydrophobic, has a variety of different functional roles. It actually has important roles in membrane structure. So when you put it into a biological membrane, it can change the properties just because it's so rigid. It's the metabolic precursor to a variety of important molecules, maybe most importantly to steroid hormones like estradiol and testosterone. And finally, it's an important integral role in fatty acid transport. It helps move fatty acids around by being a rigid brick that sort of helps solidify or rigidify those floppy fatty acids. Just to sort of put this into some more chemical perspective, what I'm doing here is showing the degrees of freedom of a fatty acid here on this side, on the left side of this. And on the right, I'm showing the cholesterol. And hopefully what you appreciate is that cholesterol is much more rigid as a molecule and has a higher number of carbons and hydrogens relative to oxygens relative to the fatty acids. Okay. There we go. That was our survey of lipids. So again, there are these hydrophobic molecules that are derived from acetyl-CoA that have a variety of functional roles. What about carbohydrates, our next class of molecules? So carbohydrates are molecules that have a formula, unlike where we had to define lipids metabolically. We can say, no, a carbohydrate is defined strictly as something that has the following formula: carbon, water to the N, where N is just the number of carbons. In a carbohydrate, one carbon has a double bond, so it will either be an acetyl, well, not an acetyl. Wow, it will either be an aldehyde or a ketone. And that specifically the aldehyde will be in the 1 position and the ketone will be in the 2 position. So basically the ketone, the double bond has to either be here or here at position 1 or position 2. The rest of the carbons all have a hydroxyl hanging off of them. Any molecule that meets all of these criteria is a carbohydrate. So what I'm showing here on the left is the carbohydrate D-glucose, which is extraordinarily important carbohydrate we'll encounter a lot in this class or these set of videos. And so that's, you can look at it. You can appreciate its glory because you're going to see a lot of it. But the thing about, we can look at glucose and ask, does it meet these criteria? And so what we notice is it has an aldehyde at the top, position 1. And then if we look at the rest of the carbons, hydroxyl, 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 hydroxyl. So this, fortunately for us, glucose remains a carbohydrate. So carbohydrates are used for a bunch of different things. Maybe most commonly, at least we talk about in biochemistry classes, it's used as a fuel source. So it's the starting point for a whole lot of energy consumption in cells. It is a metabolic precursor. Carbohydrates are metabolic precursors for a variety of different molecules, and then also plays structural roles. So for example, plants polymerize carbohydrates to make cellulose, which obviously is a rigid material that's used in wood. Now, at least in humans, biological carbohydrates range from 3 to 7 carbons. So I'm just showing examples of those here, going from 3 to 7. But due to the chiral centers, there's actually a lot more than just 7, or, we could just look at this naively and say, oh, look, 1, 2, 3, 4, 5, there are only 5 possible sugars. But because there are all these chiral centers, there's actually multiple ways to be each of these sugar types. So for example, if we look at 3 carbon sugars, there are 3 different enantiomers possible. So we've got, I'm actually showing them all here: D-glyceraldehyde, L-glyceraldehyde, and dihydroxyacetone. You can look at the difference here, right? D is where is this hydroxyl, L is where is this hydroxyl, and then dihydroxyacetone is where is the, is it an aldehyde or a keto, right? So there's 3 different ways to be a 3-carbon sugar. As you think about the combinatorics, right? If you have a 4-carbon sugar, you actually have more ways to arrange it. So there's actually 6 ways to be a 4-carbon sugar. If you make a 5-carbon sugar, there's 12 ways to do it. And I don't want to draw them all, so I've only drawn 2. If you have a 6-carbon sugar, you now have 24 different enantiomers. And if you have a 7-carbon sugar, you've got 48, right? So there's just this huge number of different ways to be say, 6-carbon sugar. And a lot of biochemistry is transitioning between a small subset of these. Like, we don't actually tend to biologically make all 24 enantiomers, but we make a good chunk of them. And so in a biochemical context, we'll encounter like, oh, a glucose turns into a fructose, which is a movement of, say, from an aldehyde to a keto, for example. So what the idea here is not to get lost in the details for the moment, but to know that there is a bunch of complexity that we will encounter in situ as we start working through metabolic pathways. Okay, so carbohydrates in biology, in humans at least, tend to be 3 to 7 carbons long. There's a bunch of different chiral centers, so there can be multiple different forms of each of those. Adding another layer of complexity, even a sugar that has an identical, one of these sugars, so here I'm just drawing D-glucose again, can exist in multiple different conformations. So here I'm showing D-glucose in its open form, so as a linear chain. This is certainly something that's populated if you just put glucose into water. But it can also form rings. And so here are the ring forms of this. This is what's known as a furanose form, because it's a 5-membered ring. And what we're seeing here is basically a ring closure event happening between the 1 position and the 4 position that makes this 5-membered ring and ends up either putting the oxygen from the aldehyde below the ring or above the ring, alpha or beta. So glucose, 1 linear molecule, can form 2 different versions of the 5-membered ring depending on the chirality of this hydroxyl. And just to make it even more fun and complicated, it can also form a puranose form, which is a 6-membered ring. That's a connection between position 1 and position 5. So that's, different ring structure. And again, the hydroxyl can either be in the alpha form, which is below the ring, or in the beta form above the ring. This 1 carbon, this 1 glucose, this 1 type of sugar, you put it into water, it will just flip back and forth between all of these different conformations. Fortunately for us, this is the preferred form of glucose, so when you see it drawn, we'll almost always draw it in this form, this 6-membered, this puranose form of glucose. Okay, again, the point is not get lost in these details, memorize them. The point is, later on when we start encountering sugars, you'll say, I've seen that before somewhere. So planting a seed in your mind, putting maybe a mental space where you can start hooking on details as we talk about metabolic pathways later. Last complexity with sugars is they can also polymerize. So what I'm showing here is a carbohydrate, glucose, that is bound to another glucose covalently. This covalent bond is known as an alpha 1,4 bond because it's between the 1 position of 1 carbon of 1 sugar and the 4 position of another. This can just go on forever, and we can make a whole bunch of alpha 1,4 bonded sugars, making a long linear chain. And just to make it even more exciting and complicated, we can also form branched chains. So here we've got an array of glucoses that's 1,4 linked on this axis, and then here's a 1,6 link that pops off the other side. Now you might say, oh my goodness, why in the world do we have all that? That's just a ridiculous complication. But it turns out this ability of sugars, of carbohydrates to polymerize is really important. So when we talk about glycogen, that's the way that our cells store sugar for later. They do so by building branched arrays of sugars, branched arrays of glucose. Okay, so that was a lot of content, a lot of complicatedness. This is really the paradox of carbohydrates. In some ways, they're like the simplest, right? You're just like, oh, it's just a carbon with some water, the simplest biomolecule. But they can be assembled in so many different ways that even practicing biochemists like myself, if I ever encounter carbohydrate chemistry as part of my research, I sort of tense up because whatever we're going to do is going to be very complicated to deal with. So if you're feeling overwhelmed by carbohydrate chemistry, I guarantee 99% of biochemists that like do this for a job or also feel overwhelmed by it. So you're in good company. Okay, so we've talked about lipids, we've talked about carbohydrates. Let's talk now about amino acids. So all amino acids have a common structure, and that structure consists of an amine on one side, a carboxylate on the other, and these are bound together in the middle by an alpha carbon. This alpha carbon has a proton that hangs off of it, as well as an R group. And this variable R group defines the identity of the amino acid. So this is variable, as is typical, in an organic chemistry context. When you see an R, right, it's just like, it's something. So it turns out with an amino acid, it's something. Amino acids are used maybe most famously for protein structure, but they also play critical roles in nitrogen metabolism, which we'll unpack later. And then they can actually be used as a fuel source as well. Now, I'm going to very briefly talk about their role in protein structure here, but I've got a whole stack of videos planned for this that you'll get to look at in more detail later. So again, we're just going to go over it at a very high level. If we look across cells, we have 20-ish we have 20 amino acids that end up in proteins. There's a few more that end up as part of metabolism. Like I said earlier, amino acids are defined by having an amino side and a carboxy side. If you look across this big set of amino acids, you'll notice that they all have an amino side and a carboxy side, an amino side and a carboxy side, right? That's repeated. But then they differ in their R groups. So the R group of the amino acid alanine is a methyl, just a CH3. Whereas, say, the R group of a cysteine is a CH2SH. That's its R group. You'll recall earlier I mentioned, this is the sulfurs sprinkle into amino acids. Turns out they sprinkle into the amino acid cysteine. Come on, laser pointer. There we go. The amino acid cysteine and the amino acid methionine, wherever that is, methionine. That's where our sulfurs come. And again, we're going to do a whole set of videos about how these amino acids interact to define protein function or proteins themselves, but I'll briefly mention two features of them. The first is that because these amino acids have this repeating structural element of an amino side and a carboxy side, they can be assembled into a chain of amino acids of different identities, right? So you can just sort of glue them together end to end. This forms these bonds here known as peptide bonds. Whoa, yep, that is not a peptide bond. That's the peptide bond, sorry. It forms this array of peptide bonds, which then allows this chain of amino acids to fold up into particular structures. And so when we get into it, we're going to talk about a whole bunch of different protein structures. These are defined by the amino acids that are present. And again, since we're going to spend so much time on that, I think I'm going to leave amino acids for now and move on to our last class of molecules, nucleic acids. So, okay, lipids, carbohydrates, amino acids. Finally, let's talk about nucleic acids. So nucleic acids have a stereotypical structure as well as just like amino acids. That stereotypical structure consists of a phosphate here, a ribose sugar here, and a ribose sugar is just a 5-member ring with a specific, actually there's a specific enantiomeric state of all of the hydroxyls. And then what's commonly referred to as a base. And this is what determines the identity of each nucleic acid. So this would be the equivalent of an R group in an amino acid. You have an R group for each of your nucleic acids. Nucleic acids are used possibly most famously as the genetic carriers in both DNA and RNA. But they also have important roles as in energy currencies. So ATP is a nucleic acid. And then they're used as metabolic precursors for a whole bunch of different molecules. So they have a wide variety of roles, which we'll encounter throughout this set of videos. These are the 5 most common nucleic acids here. And hopefully what you can see is they all have a shared architecture down here with a phosphate and a ribose sugar. And then this one over here is a little bit of a weirdo. This is actually a deoxy form. So this would be what we see in DNA, whereas these are on the oxy form, which is what we see in RNA. I realize I'm kind of hanging out over the bottom base, so I'll make myself disappear for a second. These bases, this is adenosine up here, which pairs with uracil. And this is guanosine, which pairs with cytosine. And then finally, this one over here is a thymine base. We'll talk about these later when we get into nucleic acid chemistry. But just so you know, each of these have a name, just like, when we say, oh, we've got a nucleic acid sequence of A, G, C, T, we're talking about a molecule that's got an A bound to a G, bound to a C, etc. Adenosine, it actually turns out occurs a lot. We see it all across metabolism. And so I'm showing the structure of the molecule here. Most common place it probably appears is here in ATP, which you'll notice is just an adenosine base with two phosphates hanging off the side of it. It also, though, gets modified. So, for example, here, this is cyclic AMP, an important signaling molecule. And hopefully what you appreciate is this is an adenosine with a slight modification, a cyclicalization between the ribose sugar and the phosphate. But, oh, looky here. Remember this molecule? We saw this earlier. This is the CoA part of acetyl-CoA. Notice this part. That's just adenosine base that's stuck onto a bunch of other stuff. This molecule here at the top, NAD+, we're going to encounter a lot. This is important in electron sort of redox chemistry. Look, it's an adenosine base. And here's FAD, another important molecule in electron transport. Oh, look, it's got another base, right? So adenosine in particular is one to keep track of and pay attention to because it's going to appear all across biochemistry in these various guises. Okay, so let's quickly walk through each of these classes. Again, I know this was a lot of content, sort of a data dump, but I thought it was useful to put it all together so we could sort of compare and contrast all of these classes of molecules. So we've got lipids, which are used as fuel sources, energy storage, and signaling, and they tend to be these greasy hydrophobic molecules with lots of carbons and hydrogens. We have carbohydrates, which are also used as a fuel source, but are they act as metabolic precursors, and they can be used in structure. And they tend to be characterized by CHO. We've got amino acids, which play roles in protein structure, nitrogen metabolism, and then they can eventually, if we run out of other things, be used as a fuel source. These have CHON and then a little bit of S. And then finally, we can talk about nucleic acids. We talked about nucleic acids, and these are used in RNA and DNA. They're used as energy currencies, and they're used as metabolic currencies. And these have CHONP. So as we go through this set of videos, as we start covering things like metabolism, we're going to start encountering all these molecules, and hopefully this provides a grid to at least sort of say, okay, I've seen a carbohydrate before, or I've seen a nucleic acid before, as we dig into those more detailed processes. But I would strongly encourage you, as we go through stuff, as we talk about details, to continually look for patterns instead of memorizing details. If you have a half-empty approach and you're like, I've just got to keep track of all the stuff, you might learn some things. Maybe that's your learning style. I don't want to, crimp your style and say don't do that. But I think overall, for most students, you're going to be much more successful if instead you say, all right, if I know a few core things well, if I can see these patterns across biochemistry, you'll be in a much better place to understand what's going on as we go into all the crazy details here. So, all right, so that's what I have for this. In the next video, we're going to talk about the sorts of molecular interactions that can form using some of the molecules we've already talked about as examples. I look forward to seeing you there.